Hi everyone. Let me open my presentation. Okay, hi everyone. My name is Catherine Crabtree, and today I will be talking about the chapter 2, and I'm just going to kind of touch some of the main points right here. So, first and foremost, for the just kind of the overview of what we're going to be talking about today, we're going to be talking about properties of light, microscope developments and comparisons. What is microscopy? Instruments of microscopy, staining of microscopic specimens, why is staining important, and types of staining and their purposes. Okay, so for production of light, we have the wavelength, we have the amplitude, and we have the frequency. So wavelength is the distance between peaks, amplitude is the heights, and the frequency is how close these wavelengths are together or how far apart. So whenever we have a lower frequency, we see the wavelengths being super far apart. And whenever we have a higher frequency, we see these being super duper close together. Some microscope developments and comparisons. So the creator was actually Antony van Leeuwenhoek, I think is how you say that name. Although some believe credit should be given to the lens crafters or to actually Galileo, we do still credit Antony van Leeuwenhoek for that initial accreditation. And speaking of Galileo, he actually used a compound microscope, whereas Leeuwenhoek only used a simple. Whenever we're talking about compound versus simple, we kind of think compound is more complicated, is how I like to think of it anyway. And simple is simplified. So compound has 2 and simple has 1. Some early discoveries that were found with the microscope was actually found with the compound microscope, and Hooke used a compound microscope to observe a sample of cork that resembled a honeycomb. What is microscopy? Microscopy is a way to for us to kind of see these tiny objects, like cells, bacteria, tissues, or other structures that are too small to be seen without magnification. We can't see them with the naked eye. We can't see them with our glasses on. We can't even see them with those really magnified readers on. Not happening. So instruments of microscopy. We have light microscopy. This uses light and lenses to magnify objects. It is the most common type of microscope. I remember, if I'm correct, using a light microscope in anatomy to see veins, venules, different types of cells, epithelial cells, see different types like eukaryotic, prokaryotic cells. I remember using that. We also have electron microscopy, excuse me. This uses electrons to create highly detailed images of very small structures. The beams in the electrons uses beams of electrons instead of using light, because electrons have shorter wavelengths, so they are more defined and reveal like much finer details. And then we also use a scanning probe microscopy. These use tiny probes to scan a surface and get a detailed image, and it can even see individual atoms. So it is a very in-depth, defined microscope. So we're going to move on and to staining of microscopic specimen. So wet mount preparation. This is kind of more of the preparation side and the initial staining. Wet mount preparation is whenever we take our specimen and we put it onto a like a drop of liquid. Fixation preparation, it can be more of it can be heating or chemically treating the specimen. And staining, it colors certain parts of a specimen, either the background or the specimen itself. Why is staining important? I feel like staining is something that we see a lot. We've seen it in anatomy, we've seen it in, whenever we did like lymphatic vessels, we saw it using staining in order to see like what is a lymphatic vessel. We saw it with granulocytes and agranulocytes. So why is it important? It's because many cells are transparent. They are difficult to see under a microscope, and the stain, it adds this contrast. So whether it stains the background or it stains the cell itself, you are getting that contrast in order to be able to see the subject. And it helps make those structures stand out. It can help us see the nucleus, cell walls, bacteria, and different tissues. So we're going to be talking about different types of staining and their purposes. So we have gram staining, acid-fast staining, capsule staining, endospore staining, and flagella staining. So for gram staining, it detects cell wall structure, and it differentiates bacteria into gram positive or gram negative. For acid-fast stain, it detects that waxy cell wall. It identifies like acid-fast bacteria, like those that cause tuberculosis. Then we see the capsule staining. Capsule staining, it detects capsules and it reveals a protective capsule surrounding some bacteria. So some bacteria may be harder to break down. And then we will look at endospore stain. So endospore stain, it detects endospores and it shows bacteria that can form resistant spores. So I work in an urgent care, and whenever patients come in with UTIs, we will sometimes send them off for what we call a culture. And they take it to the lab and they test it. And they can see what this certain bacteria is resistant to or what will work with it. So we are actually able to see what antibiotic we can give a patient that may have Klebsiella form a UTI, or they may have a staph form a UTI, or an E. coli form a UTI. And sometimes those antibiotics are different depending on what kind of UTI a patient has. And flagella stain, it detects flagella and it makes bacterial flagella visible. Lets us know what's really happening. So in conclusion, we have talked about the creator of the microscope, whether we want to give that to Antony van Leeuwenhoek, or if we want to give it to Galileo, it is subjective. Or if you want to give it to the lens crafters, it is subjective. We accredit Antony van Leeuwenhoek, but many people argue it. We've talked about compound versus simple. We know the difference. We should know the difference now. Compound, complex, 2. Simple, singular, 1. We know that Hooke used a compound microscope to find the findings of the honeycomb. We know light microscopy. We know electron microscopy. We know scanning probe microscopy. We know what microscopy means. We know why staining is important. We know the different types of stains: gram stain, acid-fast stain, endospore stain, flagella stain, and capsule stain. And that is the end of this presentation, guys, and I hope we have enjoyed, and I'm glad that we have got a good understanding of chapter 2.